Hello everyone, my name is Anjali Tanatur Vinod. I'm currently a master's student in Yonsei University, Department of Physics, and I would like to talk about my research proposal, which mainly focusing on developing a photonic chip for contactless particle manipulation. So I'm really excited to share my uh, research proposal in the field of photonics, specifically the development and application of all fiber basal beam generator or BBG in advanced microscopy and material handling. We aim to experimentally demonstrate the multi-channel optical microparticle transport using BBGs embedded in a weak groove fiber array chip using a commercial fiber coupled laser diode. So let me move to the PowerPoint. When you see the outline, it starts with introduction. We briefly introduce the concept of our research and methodology which we would like to implant, then expected outcomes, timelines, significance of our research, and budget at the end, the conclusion. So let me begin with introduction. So, welcome to our exploration of bezel-like beam traditionally used in advanced microscopy and material handling through expansive bulk optic platforms. Our study pivots towards a novel approach using cost-effective all-fiber bezel beam generator within multi-mode fibers. BBG in action, showing a non-diffractive properties and microparticles transport. This innovative beam generator showcases unique non-diffractive properties, enabling contact-like microparticle transport with high-power lasers. A critical application of this technology is in fluid dynamics. Our study concentrates on understanding these beams in aqueous environments. We aim to demonstrate multi-channel optical microparticle transport using V-groove fiber array chip powered by a commercial laser diode optimized in D2O medium. The insight gain could pave the way for advanced integrated photonic chip employing multiple bezel beam generator, revolutionizing the optical microparticle manipulation. So let me move to the methodology of our research. So our innovative research seeks to create an all fiber bezel beam generator. We begin by precisely fusing a single mode fiber with multi-mode fiber to generate optimal bezel like beam at a wavelength of 976 nanometer. The reason why we use 976 nanometer because mainly it's the cheapest available fiber coupled laser diode. So when you are doing a research, you should consider about the expenses of the equipment. So I really wanted to put my all effort on it. Through careful alignment and fusion, we achieved a beam whose transverse intensity distribution mirrors the desired Bessel function. This is verified using CMO sensor for real-time observation. Moving forward, our experiment entails the parallel array trapping of 10 micrometer polystyrene microspheres, showcasing the precision of our BBG system. So I would like to start with a 10 micrometer particle, which is the size is similar to the biological molecule. So I would like to implant this method to in biology. We'll also would like to delve into the photothermal effects and conduct a beam profile simulation, further demonstrating our system's versatility in different mediums. D2 is the medium for our trapping. D2, known for its low absorption coefficient, is our chosen medium for trapping. We will validate its efficacy using COMSOL simulation. In conclusion, our methodology paves the way for advanced microfluid applications, harnessing the power of basal light beam for contactless particle manipulation. So the next slide, I would like to briefly describe the my experimental setup which I would like to do experiment with. So these are 3 laser diode. I want to connect it with 3 bezel like beam generator and arrange the fiber parallelly using a V-groove fiber array. I would like to hold the fiber using a PDMS crystal which make the fiber like motionless, like fiber should be fixed and pass the laser through it, and would like to observe the particle trapping, or like bezel like beam particle trapping using a CMOS camera sensor, as you can see in this figure. So the best research you can do is to talk with the people. So you have to collaborate with different research institutes in order to gain knowledge about your research, and you ask the question about what you want to do. So the research equipments I needed for this experiment are like, initially it's like optical microscope, which is available in our lab, and laser diode, then heavy water D2O, which is kind of expensive, but we need to buy it for the experiment, since D2O is the medium of trapping. uh, the next is like dielectric particle. We would like to use the polystyrene microspheres and the array holder and some other material for holding the fiber and absorbing the trapping, and media beam profile analysis. So these are really important. So when you when I would like to talk about the expected outcomes, the research led to the development of innovative all fiber bezel beam generator, a significant advancement in photonics. uh, this generator was crafted by leveraging mode mixing interference with a multi-mode fiber at a precise wavelength of 976 nanometer. The key to its performance was the meticulous fusion of single mode and multi-mode fibers, which produce an optimal bezel-like beam. This achievement was quantitatively confirmed through experimental observation using CMOS sensor to analyze the beam's transverse intensity distribution, accurately reflecting the bezel beam pattern. Further, the research showcased particle practical application uh, of uh, of the technology through successful trapping of 10 micrometer polystyrene microspheres arranged in parallel array configuration. This not only demonstrated the generator's potential in microscopic particle manipulation, but also its precision and reliability. The team pushed the boundaries by exploring the BBG's capabilities in various environments, delving into photothermal effect and utilizing beam profile simulation to understand the full scope of BBG's performance. Lastly, the choice of D2O as a trapping medium was validated through COMSOL simulations, with results highlighting its suitability due to its low absorption coefficient in 976 nanometer wavelength. This comprehensive research provides a promising foundation for further exploration and utilization of bezel beams in diverse scientific and industrial application. The next thing you have to from September 1 to October 31, this timeline is for refinement and validation. I want to refine the research methodology based on the preliminary analysis, then validating findings through additional experiments or analysis. From November 1 to December 31, I would like to prepare the manuscript, start drafting the research manuscript, collaborate with key researchers on manuscript content. In 2024, from January 1st to February 28th, the peer review and revision. Submit the manuscript to the relevant journal, address peer review feedback, and revise the manuscript accordingly. From March 1st to April 30, I would like to participate in some conference and submit. Identify the conference relevant to the my research. Mainly, I'm focusing on Clio. Then prepare and submit conference abstract. Since conference is really important to communicate your ideas or just demonstrate what you have done in front of the scientific society, so it's really important. Then from May 1st to June 30, I would like to participate in a conference, attend selected conference to present my research, network with researchers, and receive feedback. From June 1st to August 31st, I finalize the publication and post-conference adjustment. So the points I need to consider during this research includes be flexible with timelines as a research progress may vary. Then regular communication with collaborators to ensure the alignment, and finally adapt the timeline based on actual progress and unforeseen challenges. Since it's a research, I should do everything according to some plan to get succeeded in result. The applications of and significance of this photonic chip which we developed include like particle sorting and drug delivery in human body. Also, it can replace microfluidic chips, which is really expensive, and you can remove it by replace it using our chip. Then budget is an important thing. Our initiative requires substantial funding to acquire essential optical component, establish experimental setups, and access sophisticated data analysis tools. This investment will also ensure the hiring of adapt research assistants and the provisioning of comprehensive laboratory resources. By directing funds towards these pivot areas, we aim to foster a fertile research environment conducive to the innovative and research discovery, and which is very important. And I would like to conclude my PowerPoint by the next slide to elucidate the strategic approach undertaken for advancing and understanding the practical application of all fiber bezel-like beam generator in realm of microfluidics. We delve into multi-phase research and development program. This program is very important for our further research. Systematic approach to advanced understanding and application of all fiber bezel-like beam generator in microfluidics is the main goal of our research. Then next goal is to create a versatile and integrated multi-channel optical transport system with a practical application which can be used in microfluidic channels and drug delivery, mainly in biological systems. So this is all about what is my research proposal is. Thank you for listening to me and have a nice day.